a whole lot more. And then the final stage, stage 4, is you're going to get cellular characteristics developing through what's called chemical selection. Thus far, we have talked about natural selection. That's not happening yet. We don't have active reproduction. What we have is chemical selection. All chemical selection says is the chemical that's easiest to make or that is produced in the most abundance will be found most commonly. If it's easier to make it, that's what's going to be made. You have it in very, very high quantities. So if a molecule is able to self-replicate, to build itself, to catalyze the creation of itself, that's going to be the one that is most heavily favored. And it turns out, though right now we put a lot of emphasis on DNA, it was RNA that was able to do that self-replicatory function. RNA has a lot of qualities that says it was probably the very first macromolecule in protobionts. Not proteins, not DNA, it was RNA. And we kind of regulated RNA to a secondary status now, where like RNA's job is just move a message from here to here. But RNA does a lot of things, and it's got a lot of unique attributes that make it important. Because of the way the base pairs work, it's great for information storage. You can use a piece of RNA with a single strand, and because it has that single strand with those open nucleotides, it can complement itself. That is, other nucleotides can bind to it to create a template, to create a mold that you can use to make more RNA. Those same qualities, those same base pairing qualities, mean that RNA has the capacity to reproduce itself, to replicate itself. It's sort of like a robot building a robot building a robot. RNA, once it's got a certain form to it, acts as I believe it's called a ribozyme. It's the idea that it will help to facilitate the creation of even more RNA. Oh yeah, it is called a ribozyme. Look at that. So this is a piece of RNA, twisted up on itself, self-complementary. RNA, we have always talked about as is single-stranded. The thing is, it is a single strand, but that strand can interact with itself, leading to different structures forming. You guys have pulled out a long piece of tape before, right? You know what I'm talking about, Scotch tape. Whoosh, pull it out. What happens to the tape as soon as you let go of one end? What? Okay, how about this? It sticks to itself. Oh, are you talking again and I'm not hearing? No, it's not okay. Come on, you could do this. Just speak. All right. It sticks to itself. This is like the bane of Christmas, right? You're wrapping presents, you pull out a piece of tape, it gets stuck on one end of the wrapping paper, and then you pull it off and the wrapping paper rips, and then you think to yourself, I don't know, should I change the whole wrapping paper or do I just patch over it? I assume you've done that before. You've had that thought of, do I just patch it or do I have to redo the whole thing? You just patch it. And then you're like, do I patch it just with more tape? And then suddenly it's like this big splotch of tape because you have to reinforce that section. Or do you take another piece of the same wrapping paper and try and match it? You do that. There's all sorts of things you can do. Because tape sticks to itself. RNA will also stick to itself, forming these complex structures. The deal is, if one of those shapes, if one of those forms, of all the random of strings of RNA that formed, could replicate itself, that gives it an extreme chemical advantage for selection. If any of that RNA, now this is across the entire world and all the oceans filled with these biological molecules, if any one of those RNAs was able to pick up a single mutation that allowed it to be to have a chemical advantage, it's going to be reproduced faster and start filling up the seas. So it's hypothesized that the early Earth history was an entirely RNA world. It was RNA encased in a liposome. Likely at a pile of clay on the edge of a sea. That RNA acquired the ability to enzymatically link other nucleotides. That is, it acquired the ability to build itself, self-replicatory. As that chemical selection acted on the RNA became capable of bonding amino acids together. So you have this structure, so it's not just able to enzymatically link itself, now it's able to enzymatically link amino acids. Now we know this had to happen. And the reason we know this had to happen is because that's all a ribosome is. A ribosome is RNA twisted on itself around an amino acid. So it definitely happened at some point. Don't know when, but we know it happened. And what do ribosomes produce? I'm not trying to trick you. Do you guys remember? Ribosomes make what? Did she say something and I missed it again? No? Okay. Not this time? Bio 101, big concepts, central dogma of molecular biology, absolutely crucial to all modern biology. What do ribosomes do? Yeah, there we go. Between the two of you, synthesize proteins. They build polypeptides. Let me go over how ribosomes work really quickly, because I'm not sure you're on the same page as me. The way a ribosome works is an RNA message will bind to a ribosome. Now again, a ribosome is simply RNA wrapped around a protein. That RNA message is then translated. Every 3 nucleotides is considered a codon. Each codon encodes for a certain amino acid. So get this, another bit of RNA, because can't get away from RNA, it's called transfer RNA, tRNA, is going to carry that amino acid with it. It's going to land in the ribosome. It's going to drop off the amino acid it's carrying. Those amino acids are then linked by the ribosome, by that bit of RNA, enzymatically to create a polypeptide. That polypeptide is then going to go through a hierarchy of structure, of primary, secondary, tertiary, quaternary structures, where it gets a certain shape to it, certain form, and that form ultimately will determine its function. So it had to happen. At some point, RNA definitely got the ability to link other molecules together. But is it possible for it to happen all by itself without any other intervention? Turns out it is. This is Dr. Jack Szostak. He won a Nobel Prize for this. Back in 1993, he put 10 to the 15th power. So, that's a lot of zeros. Yeah, sorry. Long strands of DNA. And they were mixed in with a very short strand. So you've got a long strand of DNA, and these are randomly created long strands and randomly created short strands. Just like conditions on the early Earth, there's no real plan to it. Absolute random. At that point, he decided to do a selection event. He put all of those long strands of RNA in with the short strands of RNA. And what he ended up finding was he wanted the long DNA to be a catalyst for binding the other molecules together. So he ran an experiment. He put them all together and mixed them up. What he was looking for was even longer RNA. So if you have a long, so let's see. I'll just do this with my arms. A long piece of RNA and a short piece of RNA. Okay? What he wanted to find was there a long piece of RNA that was even longer, had that short piece attached to it? And he found that there was. So then he cleared out all the rest. He said, okay, that particular one, that really long bit, the one that was bigger than all the others, the one that was heavier than all the others, that's the one I'm most interested in. So he took that one, and he copied it, brought it up to 10 to the 15th power molecules, reran the experiment. And what he found was, in the next time, it was even longer. Not surprising, right? Not only was it longer, but there were more of them that were longer. He ran it again and again and again. He did this experiment 10 times. Each time he did it, the RNAs were getting progressively longer and more grand. After the 10th experiment, he found that the bonding tendency, the ability of these RNAs to bond to other RNA and create longer RNA structures, was 3 million times more effective than it was at the beginning. These RNA molecules were self-replicating. It's possible for it to happen. So then why don't we have an RNA world now? Well, RNA is super cool. Why isn't it here now? Well, it turns out chemical selection favors the molecule with the advantage, just like before. RNA had the advantage, but then this new molecule came around. RNA started building a double-stranded version of itself, and that double strand gave it stability. RNA doesn't last forever. It's like it's got a fuse. It wears itself out. But DNA, deoxyribonucleic acid, is double-stranded. It's got hydrogen bonds that hold it together. So DNA now takes over the role of storing genetic information. RNA is just transmitting that information. This gives the protobionts that have DNA an advantage in that their blueprint doesn't fall apart. It doesn't fray. You don't lose any bits and pieces of it. So now RNA is filling these other roles. You got liposomes that have both DNA and RNA are successful. The DNA doesn't degrade. In fact, the DNA, when that liposome splits, that DNA can copy itself. So now you have two sets of instructions in two different cells. I'm sorry, not cells yet. Two different liposomes. Now we know RNA can make DNA because of this thing called reverse transcriptase. In fact, there's telomerase. It's a group of RNAs that will come and build off the edge of a chromosome. So it is absolutely possible for this to have happened. And of course, we also know that RNA functions, like I explained earlier, in creating proteins. So RNA can build DNA, RNA can build RNA, RNA can build proteins. RNA is pretty darn important. Peptides, real quick, how many nucleotides are there? Do you guys remember? 4, right, in DNA? 4 in RNA. That doesn't lead to a lot of variation chemically. And they're very similar to each other. Amino acids, though, there are 20 of them. And they're really different from each other. That increases variation. Now you're building these enzymes that can have vastly different abilities than the RNA. They're very, very different forms. They can have very different functions. The RNA can encode for these proteins. And not just one. It can encode for 1, 100, 200, 300, depending on the different binding arrangements. So in the end, it all comes down to RNA. And even today's world is still really all about RNA. I mean, RNA is important for protein synthesis. You know, you need to have that message. Messenger RNA is what takes that message from the nucleus out to the ribosome where it binds. It's going to be transfer RNA is going to carry amino acids. Ribosomal RNA is what's building the ribosome. Without transfer RNA, ribosomal RNA, messenger RNA, none of these things would happen. You would not have proteins, and in the end, you wouldn't have DNA. So chemical selection has led to the modern world, the central dogma, which is DNA is transcribed to RNA, RNA is translated to a polypeptide. Polypeptide folds up into a protein. Proteins do like pretty much everything. So now we've moved from this world where you've got just simple molecules. Step 1, you get your prebiotic molecules. You You get some amino acids, some nucleotides forming. They polymerize. One way or another, they're going to come together, whether in water or you exclude the water. Those macromolecules, those polymers, get surrounded by a liposome. The one that's chemically selected for does the best. Likely the chemically selected for one was RNA. As those simple cells grow and they start to use electrochemical gradients to provide themselves with energy, to create more bonds, they take on simple characteristics of life. You get the earliest of the cells. And once you have the ability to self-replicate the entire cell, you move from a chemical selective phase to a Darwinian phase, to natural selection. And that's what we talked about from all the rest of the beginning of the course. We didn't really get into chemical selection earlier. It's a little bit more complex. But we hit our looking at different alleles and changes in allelic frequency, changes in fitness levels. And the organism that's the most fit survives. Once you're at that point, almost in a blink of an eye, you get complex life from simple life. And that's where we're going next. We're going to start talking about bacteria, archaeans, and protists. These unicellular organisms that are competing with each other in order to survive in this world. But in order to get to them, we had to first build those first simple cells. So what I just told you was